Good afternoon. I'm Harshvardhan. I'm going to present on a research paper on global proteomics-based identification and validation of Thymosin Beta-4 X-Linked as prognostic marker for uh, head and neck squamous cell sarcoma. Before I proceed to the topic, I like to introduce about the authors and there were many authors involved in this research. Among them, these were the three authors. Uh, Thymosin Xingle, expertise in proteomics and cancer research, and Yun Chongling, renowned for contribution in biomedical informatics, and Michael Hisog, he has done significant work in cancer biomarkers and therapeutic targets. And these three were main responsible for deciding and supervising this study. And other than that, some of them have also helped in this research. And so let me introduce the topic. So as I mentioned earlier in the paper, we are going to see the global proteomics on head and neck squamous cell sarcoma. It is a very major global health concern derived from oral cavity, oral pharynx, and hypopharynx, and it's the fifth leading cause of cancer death in Taiwan. And it has poor prognosis with current treatments, and there is high mortality rate. And there has been existing treatments have been limited success, and necessary new markers are required for better prognosis and therapies. And planning acute treatment modalities for specific subgroups of this. So in this paper, what they did was that they took a specific subgroups of head and neck sarcoma patients, and they performed proteomic analysis. Uh, Proteomics is nothing but uh, it's a method for identifying proteins and which help in prognostic categorization of cancer. And the main objective of this paper was that to identify and validate prognostic biomarkers for head and neck squamous cell sarcoma using proteomic approaches. And their main objective was to identify the thymosin beta-4 X-linked TMSB4X as the biomarker. And they had a question that whether TMSB4X can survive as a reliable biomarker for head and neck squamous cell sarcoma. And they also analyze the expression of TMSB4X in this and evaluate its potential as a biomarker. And the methodology here they used I have mentioned in a step how they have done. So first they have used cell lines and cell culture methods. So in cell lines they use 7 cell lines: CA922, CAL27, HSC3, SAS, and SCC4, and TW1.5 and TW2.6. And they have used culture media at them, and they have used antibiotics, streptomycin, penicillin, and with 10% FPS, and their incubation control was 37 and 95 percentage, and their control was NHOK cultured in keratocide, SMF, and OMF cultured in DEM. So the protein lysates and how they did the protein lysate preparation and SDS page I mentioned. And they washed the cells with PBS and lysed in protein lysis buffer and centrifuge, and the lysate was stored in it. Then 50 mg protein was heated with sample buffer, loaded in gel, and stained with colombus blue. And in gel digestion, they restand and reduction and alkylation buffers and deaeration with trypsin. And the main proteomic method they used here too was LCMS. And one of the method I'm going to explain. So the instrument used here is linear ion trap mass spectrometry, and protein for protein identification they used mascot search engine using IPA human database, and the parameters which they used were the mass tolerance 15 ppm and 0.8 uh, Dalton, and modification was excitant carboamine crystalline and variable on oxidized methionine deamination. And they have also performed malad also, so malad IMS. So in that they prepared frozen section skirt and based on the slides and ethanol, and instrumented here use was Ultraflex MLD TOEFL and spectra occur and calibrate intervals, and immunohistochemistry was also performed, and they did for 86 patient tissues, and staining they used TMSB antibodies used and slice scans and scored for staining severity, and real-time PCR analysis also was done, and they did RNA extraction from tumor and HNSCC and detected the TMSB4X mRNA levels measured by cybercrime real-time PCR. And they also did self-row refrigeration and mobility assays. So the perforation was counted for over 5 days, and mobility was checked by Boyden chamber assay to assess the cell movement and emission. And they did xenograph mouse experiment also, so which was conducted under approved protocol, and SAS cells were transinfected with luciferase plasmids inoculated into mice, and tumors were monitored. And when they were analyzing the MS data, they used tools like flex analysis, clean pro tools, and methods. They use multivariate proteomic algorithms, Kaplan-Meier estimates, Cox regression, PSNJ square test, and they got a significance of 0.05 considered as significant lesser than this. So now I'll be explaining the results. So the first they identify the biomarkers through malad IMS. So in that they took the proteomes from different stages 1 to 3 of the head and neck sarcoma and were analyzed using pro tools, and the peaks in mass spectra were compared between normal cells and cancer cells. In that TW1.5 and Cal27 were used, and the samples were analyzed with malad IMS after HCS staining, and the intensity of the distribution of the peak at 4968 point was noted. It was the strongest intensity in the advanced cancer stage. And here in the graph, the red arrow shows where the tumor intensity is greater than the normal intensity. So here if you see in this image, they have showed the different stages of the cancer, different stages of the cancer, stage 1, stage 2, stage 3 image, which is from head and neck. And they are given the process how they have done. So malad image, clinical tools, and the potential targets, then validation, then literature, and they have used mascot also. So next, the LCMS analysis of TMSBS. So here they identify the peptide KETIEQ KQAGES from Cal27 cell line was identified as TMSB4S, and peptide sequencing was confirmed by matching B ions red and Y ions. So the B ions are the charge retained by amino terminal part of the peptide, and Y ions charge is retained by the carbo terminal in the peptide. So in this image, they have showed that the peptide K and how it has been identified, and they have showed the observance, and in that they confirm by matching V ions and Y ions in sequence. And next, they then immunostaining in HS staining in HNC tissues, and the tissue microarrays from 31 HNC samples were stained using immunochemistry, and among them, three represent pair images of normal and tumor tissues were shown. So these were the image which were done staining, and they showed three paired images from normal and tumor. So in this image, you can see the difference how the normal cells were there and how the tumor was there, and this. So this then they found out, as they have done a knockdown study in mouse model. So they have checked the cell function assay. So knockdown of TMF in SAS cells may reduce the proliferation and invasion in vitro and suppress the cervical metastasis in vivo. And the RPC, RT-PCR analysis was done in that in 6 HN cell lines with high levels, the aggressive SAS cells were seen. So in RTR expression, 6 cell lines, high levels in aggression was seen in SAS cells. So that is in the graph. And finally, they have checked the bioimmunance, the mouse model which they have done and which were taken from buccal orthopolisonograph. And here the total luminance was measured separately from the tumor in the mouth and neck lymph node. So they have checked also. And yeah, so conclusion. So in conclusion, I like to highlight the main objective of this paper. And first, it was TMSB4 expression, which is higher than HNC tumor cells, especially in stage 3 and 4. It was confirmed by HNC staining and RT-PCR. And it is a small protein that binds to actin, preventing its polymerization until it is needed for filament formation, its main function. And if TMSB4X is upregulated in various cancer, including gastric, colorectal, pancreatic cancer, it enhances the cell motility and inversion and metastasis. And it also promotes the epithelial mission transition, cell motility, and androgenesis via various signaling pathways. And knocking down the TMFB in cell line reduced cell proliferation and invasion and metastasis. And there was a reduced lymph node metastasis in mice with TMS4 knockdown was observed. And TB500 drug, which contains a segment of TMSB, which promotes human healing and it's used in doping also, could be explored for HCS treatment. And the future research, you can use Malide IMS for gene network analysis in HN tissue microarrays and develop antibiotics against this to inhibit the progression. And in summary, I like to highlight the importance as the TMSB4X is a potential therapeutic target for head and neck sarcoma, cell sarcoma, and it is due to its promoting tumor growth and metastasis. And further research could be lead to new treatments based on emerging process. So, thank you.